Let's talk about the Krebs cycle. It is also known as the citric acid cycle or the tricyclic acid cycle. It was named after Sir Hans Krebs, who elucidated it in 1937, and he even received a Nobel Prize for it in 1953. The cycle occurs in the mitochondrial matrix and turns once for each acetyl-CoA that enters. Now, since we have two acetyl-CoA molecules per glucose molecule, the cycle is going to turn twice per glucose molecule. This distinction per turn versus per glucose is one of the most common sources of confusions in examinations, so I want you to remember it carefully. The cycle begins with condensation. Two carbon acetyl groups from acetyl-CoA is joined to the 4-carbon molecule oxaloacetate, or what we are simply going to call OAA, by the enzyme citrate synthase. Write it down. This produces a 6-carbon molecule called citrate. Coenzyme A is released and recycled. This step is irreversible and commits the acetyl group to the cycle. In the second step, another enzyme called aconitase converts the citric acid, which can also be called citrate, to isocitrate, or what we call isocitric acid, via a dehydration and rehydration reaction. Essentially, we are repositioning the hydroxyl group, so no energy is released here. This is simply molecular rearrangement. In the third step, isocitrate dehydrogenase catalyzes the first oxidative decarboxylation. So isocitrate loses the carbon dioxide and is oxidized. Can you locate it on the picture? Reducing NAD plus to the first NADH of the cycle. The first carbon molecule, alpha ketoglutarate, the 5-carbon molecule, alpha ketoglutarate, is produced. This step is rate limiting. This means that it is inhibited by high levels of NADH and high levels of ATP, meaning the cycle slows down when the cell already has plenty of energy. In the fourth step, the complex alpha ketoglutarate dehydrogenase, which is a multi-enzyme similar to what we saw earlier, PDC, catalyzes the second oxidative decarboxylation. Alpha ketoglutarate loses another carbon dioxide and is oxidized to produce the second NADH of the cycle, and a 4-carbon molecule, succinyl-CoA, is produced. I want you to notice that both the carbon molecules that entered as acetyl-CoA have now been released as carbon dioxide. In the fifth step, succinyl-CoA synthetase converts succinyl-CoA to succinate, releasing coenzyme A. The energy stored in the thioester bond of succinyl-CoA drives the only substrate level phosphorylation in the Krebs cycle, generating 1 ATP directly per turn. That means 2 ATP per glucose molecule. In the sixth step, succinate dehydrogenase oxidizes succinate to fumarate, reducing FAD to FADH2. Crucially, this enzyme is physically embedded in the inner mitochondrial membrane. It is actually complex 2 of the electron transport chain, meaning FADH2 is produced right where it needs to be used. Don't forget that. In the seventh step, fumarase adds water, the double bond, across the double bond in fumarate to produce malate. Can you locate malate? This is a simple hydration reaction, and there is no energy change. In the eighth step, malate dehydrogenase oxidizes malate back to oxaloacetate, reducing NAD plus to the third NADH of the cycle. Oxaloacetate is regenerated, ready to condense with the next acetyl-CoA and begin another turn. What does this give us? pattern of the cycle, we are going to have 3 NADH, 1 FADH2, 1 ATP, and 2 carbon dioxide molecules. But per glucose, we are going to double that number. So we shall have 6 NADH, 2 FADH, 2 ATP, and 4 carbon dioxide molecules. These electron carriers, the NADH, FADH, now carry their captured electrons to the final and most productive stage of respiration. Let's talk about the Krebs cycle, the other role of the Krebs cycle. The Krebs cycle is not purely catabolic. No, far from the truth. It is what we call amphibolic. This means that it serves both breakdown and synthesis functions at the same time. Intermediates in the Krebs cycle are constantly being withdrawn to perform into other biosynthesis processes. For example, alpha-ketoglutarate is a precursor for amino acid glutamate and other amino acids via the transamination reactions. Succinyl-CoA is a starting material for heme synthesis, the iron-containing group at the center of hemoglobin. Oxaloacetate can be exported from the mitochondrion and be converted to glucose by gluconeogenesis. Citrate can be exported and used for fatty acid synthesis in the cytoplasm. Now, when intermediates are withdrawn, automatically the cycle would slow down and stop unless they are replenished. Here comes the replenishing process called anaplerosis. Say it with me, anaplerosis. The primary anaplerotic reaction is the carboxylation of pyruvate to oxaloacetate, and it is catalyzed by the enzyme pyruvate carboxylase. This enzyme is activated by high acetyl-CoA levels, which is a signal that oxaloacetate supply is insufficient. This feedback ensures that the cycle can always accept more acetyl-CoA when it needs to. Let's look at the detailed step-by-step reference of the Krebs cycle. Now, the reference slide gives you the complete Krebs cycle diagram alongside the enzymes, the products, and a quick reference table. Now, I want you to study this cycle and pace it. I want you to name each intermediate, the enzyme on each arrow, and the product of each step. Now, as you're revising this, I don't want you to copy it per word, but I want you to see it and imagine it and begin to put each enzyme where it needs to go. The table on the right is your cheat sheet. As you can see, step 1, we have citrate synthase, which produces citrate. Step 2, we have aconitase, which produces isocitrate. Step 3, we have isocitrate dehydrogenase, which produces alpha-ketoglutarate, and so on and so forth. When you're revising, I want you to challenge yourself. Cover the enzyme column and name the enzyme from the substrates and the products alone. Then, cover the products and predict them from the substrate and the enzyme. Doing this is what we call active recall, and it is far more effective than passive rereading. 2 common examinations to practice is what is the significance of oxaloacetate being both the starting and ending molecule. Do you think you know the answer? Pause and think about it. If you did not pause, this is the answer. The regeneration of oxaloacetate allows the cycle to run continuously. If oxaloacetate were to be consumed, the cycle would come to a halt. The second question: why are both carbon dioxide molecules released by steps 3 and 4 rather than at other steps? Pause and think about the answer. Both carbons from acetyl groups are released as carbon dioxide at steps 3 and 4, though due to necessary molecular shuffling, they are not necessarily the exact same two carbons that entered, but it is equivalent. Because we put in 2, we must therefore lose 2. Let's talk about the electron transport chain. This is the most complex slide, so we are going to take our time. The electron transport chain is the final and most productive stage of aerobic respiration. In fact, this is where the electrons NADH and FADH2 from glycolysis, the link reaction, the Krebs cycle, finally surrender their electrons. The electron transport chain, or what we are going to be calling ETC, is embedded in the inner mitochondrial membrane, and it consists of 4 large protein complexes numbered 1 through 4, plus 2 electron carriers called ubiquinone, also known as coenzyme Q, and cytochrome C. Complex 1, which we are calling NADH dehydrogenase, accepts electrons from NADH. As electrons pass through complex 1, 4 protons are pumped from the matrix into the intermembrane space. Don't forget that the protons are coming from the matrix of the mitochondrion and they are going into the intermembranous space. The electrons are then transferred to ubiquinone, which is a mobile electron carrier in the inner membrane. This carries them to the 3rd complex. Complex 2, which is succinate dehydrogenase, is the same enzyme as step 6 of the Krebs cycle, and it accepts electrons from FADH2, passing them down to ubiquinone. Critically, complex 2 does not pump any protons. This is why FADH2 produces less ATP than NADH. Its electrons entirely bypass complex 1. Complex 3, cytochrome BC1, accepts electrons from ubiquinone and transfers them to cytochrome C, pumping 4 more protons into the IMS via the Q cycle. Mechanism. Cytochrome C is a small water-soluble protein that is on the extracellular surface of the inner membrane. It carries electrons one at a time from complex 3 to complex 4. Complex 4, what we are calling cytochrome C oxidase, is the final or terminal enzyme. It accepts electrons from cytochrome C and passes them to the final electron acceptor, molecular oxygen. 4 electrons combine with 4 protons and 1 molecule of oxygen to produce 2 molecules of water. I said 4 electrons combine with 4 protons and 1 molecule of oxygen to produce 2 molecules of water. This is why we breathe oxygen, not to burn fuel directly, but to accept electrons at the end of that chain. 2 more protons are pumped into the intermembranous space. Now, here is a crucial connection to ATP synthesis. As the electron transport chain pumps protons from the matrix into the IMS, a massive electrochemical gradient builds up. High proton concentration in the IMS, low in the matrix, plus an electrical charge difference. This is called the proton motive force. Write it somewhere. We have said a high proton concentration in the IMS, but low in the matrix, plus an electrical charge difference. This is called the proton motive force. Complex 5, which is ATP synthase, provides the only route for those protons to flow back into the matrix, and as they flow through the F0 subunit, they drive the rotation of the molecular rotor. This rotation causes conformational changes in the F1 catalytic head, driving the synthesis of ATP from ADP and inorganic phosphate. Approximately 3 protons are required per ATP molecule synthesized. Now, I've just remembered, if you have not watched the video on the ATP synthesis structure, please watch it in the videos attached to this playlist. This entire process, proton pumping by the ETC, followed by ATP synthesis driven by proton flow, is what we call chemiosmosis, and it was first proposed by Peter Mitchell in 1961, earning him a Nobel Prize in Chemistry in 1978. Let's talk about the proton motive force. PMF has 2 components. The first is the pH gradient. The IMS has a significantly lower pH than the matrix, and this is because protons are positively charged hydrogen ions, and as they accumulate in the IMS, it makes it more acidic. The second is the electrical potential difference. The matrix carries a net negative charge relative to the IMS because positively charged protons have been pumped out. Together, these 2 gradients constitute the proton motive force, and the energy stored in this gradient is substantial, approximately 200 millivolts across the inner membrane. This electrical potential is one of the largest in biology, comparable to the charge across a nerve cell membrane, but concentrated across a much thinner membrane. Uncoupling agents are molecules that collapse the proton gradient without ATP synthesis, allowing protons to leak back into the matrix through the membrane itself, bypassing ATP synthesis. The energy is released as heat. Brown adipose tissue, the brown fat of newborns and hibernating animals, contains a natural uncoupling protein called thermogenin, or what we call UCP1. When it is activated, brown fat generates heat rather than ATP, keeping neonates and hibernators warm. Now, industrial uncouplers like dinitrophenol, or what they call DNP, were used as weight loss drugs in the 1930s. They worked wonders. People lost weight. However, they had a terrible side effect. Several people died of hypothermia because their metabolic rate spun out of control. The story of DNP is a vivid lesson in the dangers of meddling with fundamental cellular mechanisms. Let us look at a visual image of the electron transport chain. As you can see, the electron transport chain and ATP synthesis structure that are detailed in the cross-section diagram. I want you to look at the 5 complexes. Look at the mobile carriers and the direction of both electron flow and proton pumping. In the second image, I've tried to illustrate ATP synthesis in detail. The F0 subunit is the membrane-embedded rotor. It physically spins as protons flow through it, like a water wheel turned by a river. The gamma stroke then transmits the rotation the F1 catalytic head, where the alpha and beta subunits alternately change shape to bind ADP and phosphate. They then release the newly synthesized ATP. This is one of the most beautiful molecular machines in biology. Well, let's look at the ATP tally. From glycolysis, we had 2 ATP at substrate level, plus 2 NADH, which will yield approximately 2.5 ATP each through the electron transport chain, giving 5 ATP from glycolysis in total. From the link reaction, we have 2 NADH, giving 2.5 each, giving us 5 more ATP. From the Krebs cycle, we have 2 ATP at substrate level and 6 NADH, giving 2.5 each, making a total of 15 ATP. 2 FADH, giving us 1.5 each, giving us 3 more ATP, giving us a total of 20 ATP from the Krebs cycle. Put this all together will give us a theoretical approximate of 30 to 32 ATP per glucose molecule under aerobic condition. I want you to compare this with just 2 ATP from anaerobic glycolysis alone, and the importance of oxygen becomes starkly clear. Aerobic respiration is 15 to 16 times more efficient than anaerobic respiration. One important clarification: the theoretical maximum often quoted in all the textbooks is 38 ATP. However, the actual measured yield is closer to 30 or 32. This discrepancy exists simply because protons slowly leak across the inner membrane even without passing through ATP synthase, because energy is also used to transport ADP and the phosphate into the mitochondrion, and because the coupling efficiency of the F0 and F1 motor is not 100%, the lower number is physiologically the realistic figure. The next time, we are going to talk about cyanide poisoning and how it disrupts the electron transport chain. Thank you for watching. This is Educator Fresh. Please like and subscribe.